Welcome to video 11 on a cluster of technologies which has been labeled reprogenetics by some important philosophers. Reprogenetics is a cluster of genetic and reproductive technologies that have the capacity to radically reshape society. The word is a combination of the terms reproductive technology and genetics. These terms refer to two kinds of technology that we now have that were previously just science fiction. On the one side, we are able to control the process of reproduction, starting with IVF, our ability to create human life in the laboratory. The ability to culture human embryos in a test tube enables all sorts of other things. We can now have a surrogate parent, so the birth mother will be someone different than the genetic mother. We can break open the embryo to harvest its valuable stem cells. We can create an embryo that is a genetic copy of someone else. On the other side of the reprogenetic coin, we are able to control our genetics, giving us power over what it is we reproduce when we reproduce in the lab. We can select the gametes or embryos or fetuses we want based on their genes. Or we can go in and change their genes. Together, reproductive and genetic control give the human species a level of power which no one really understands yet. This control includes not only control over reproduction, but spin-off technologies that can alter the lives of adults, such as stem cell therapy and genetic medicine. We will be covering all of these innovations together because they are interrelated both technologically and morally. So here is an overview of the presentation. First of all, to understand current efforts to control reproduction, we need to understand what has been done in the past. So my first section will look at two important social movements from the 19th and 20th centuries. The first is the eugenics movement, which was an attempt to control human breeding. The second is the reproductive rights movement and the rise of the idea of reproductive autonomy, the idea that a person has the right to decide when, how, and whether to have children. With that established, I will look more closely at the enabling technology behind all of this, in vitro fertilization. IVF is a process of mixing sperm and egg in a test tube, allowing the embryo to grow for a while, and then implanting it in a woman's uterus. This is a technology that is quite familiar to some people, but its role in reshaping what is possible in human reproduction is not always appreciated. IVF gives us the repro part of reprogenetics, control over the process of reproduction. Our knowledge of genetics gives us the genetics part of reprogenetics, control over exactly what it is we are reproducing. So in the next part of the presentation, I'm going to cover the most basic application of genetic knowledge, genetic testing, and genetic screening. These technologies do not always affect reproduction, but they involve ethical issues that are deeply entwined with the rest of reprogenetics, so we need to study them now. The two basic technologies of reproductive technology and genetic technology come together in pre-implantation genetic diagnosis, where embryos are created in vitro and then screened on the basis of genetics. Of course, once we're in the business of sorting embryos according to their genetics, we might as well start tinkering with genetics, which is the next topic of our conversation. In this section, we will also look at ways of altering genes that don't involve reproduction, but can simply cure disease. After that, we will talk about an IVF spin-off that is not directly involved in reproduction at all, but rather is used to cure disease, stem cell therapy. Now, after that comes a raft of more exotic technologies, stuff that seems like science fiction, but is either real or will be real soon. This includes cloning, parthenogenesis, and the creation of chimeras or mosaics. Finally, we will talk about the ethical issues involved with the final step of the IVF process, the beginning of the business of putting a human embryo in someone's uterus, gestational surrogacy. We will then close with a discussion of general ethical issues that appear at all stages of the reprogenetic process. I think the picture you will get out of this is of a whole new kind of human reproduction, a kind of reproduction that will wind up affecting everyone, even if you never use it yourself. So let's look at the historical background of eugenics and reproductive autonomy. If you're going to talk about reprogenetics, you need to talk about the ugly history of genetics and attempts to alter the human genetics. And that ugly history is simply eugenics. Eugenics was the practice of attempting to improve the human gene pool. Basically, prior to World War II, it was common for people of all political stripes to believe that the human gene pool should be improved by selective breeding. Liberals believed this, conservatives believed this, religious people believed this, secular people believed this. It just made sense to everyone prior to World War II to say, of course, humans should be bred to improve ourselves, the way we breed dogs to get better dogs and horses to get better horses. It wasn't until the Nazi experience that people saw this as a bad thing. From 1865 to 1945, eugenics was an organized movement spanning the whole globe. Perhaps the most dramatic example of eugenics prior to 1945 was the Fitter Family competitions at state fairs. These days you go to a state fair and you see a prize pig and a prize cow and you compliment some member of the Future Farmers of America on her nice rabbits. It used to be prior to 1945 that there would also be a family there and you would compliment them on their good job of producing fit children. This makes a certain amount of sense when you consider that the impetus for eugenic ideas, wherever they crop up, is always people's success in breeding animals for agriculture. There were eugenics science foundations, and a lot of what we now think of as ordinary science was actually sponsored by eugenicists. This includes not just genetic science, but the science of statistics. All of the founders of statistics as a branch of mathematics were eugenicists. There was propaganda full of dramatic warnings about the breeding of the unfit. And this is always directed at blacks and recent immigrants, always. So when you talk about, for instance, forced sterilization, blacks are far more likely to be targeted for forced sterilization than whites, as are recent immigrants. This puts different eugenics groups at odds at each other. So an American eugenics group, say, in 1865, would be very concerned about all the Italian immigrants and what damage they're doing to the American gene pool. Meanwhile, in Italy, there's a eugenics group that is very concerned about the immigrants from the Balkans. The ubiquity of racial and national bias in these movements showed how irrational they were. All research on IQ and IQ testing is the result of the eugenics movement. If you've taken an IQ test, you've already taken a test that is descended from the tests that were originally designed to separate the genetically fit from the genetically unfit. A huge number of common tests people today are actually descended from IQ tests put forwards by the eugenicists. This includes the SAT. The whole business of standardized testing came out of the eugenics movement. The most frightening thing that came out of the U.S. eugenics movement was forced sterilization. Hospitals and prisons could legally sterilize people they felt were unfit, and often did. Around 65,000 people were sterilized against their will in the U.S. This gross violation of reproductive autonomy is now regarded as a crime against humanity by international law. Similar sterilization programs existed all over the world and probably represent the worst crime of the eugenics movement prior to the Nazi genocide. The Holocaust made all the ideas of the eugenicists remarkably unpopular. This picture captures the sudden revelation of horror when the death camps were liberated, as U.S. Senator Alban W. Barkley tours Buchenwald. The Nazis' first mass slaughter, you should remember, though, was not of Jews, but of the disabled. The T4 euthanasia program began in 1939 and is estimated to have resulted in the murder of 275,000 physically and mentally disabled people. One of the biggest U-turns in people's thinking in the history of the 20th century was the U-turn on thinking about eugenics. It went from something that everyone accepted to something that no one would even talk about. They were just embarrassed that they'd ever been involved with it. Societies and journals changed their names. Histories got brushed aside. Here we see the Eugenics Quarterly, which wasn't shamed into changing its name until 1968. We can't brush this history aside, though. It's a specter that hangs over any current attempts at genetic engineering. One of the interesting things about eugenics and genetic engineering is that the eugenics movement was focused almost entirely on removing negative traits. This is what Munson calls negative eugenics. We can define negative eugenics as the attempt to decrease the number of undesirable or harmful genes in the gene pool. Positive eugenics, then, is the attempt to increase the number of desirable or beneficial genes in a gene pool. Notice that these practices are always done on a group, on a gene pool. You cannot practice eugenics directly on an individual. In any case, the idea of encouraging the breeding of positive traits wasn't really discussed much by the classic eugenics movement. However, these days, when people talk about genetic engineering, they are more willing to accept genetic engineering to remove negative traits. After all, you are trying to cure disease, probably once and for all. The kind of genetic engineering that people are afraid of these days is enhancement engineering, which they think will create some sort of genetically engineered Superman. And although this sounds very Hitlerian, it isn't a big part of what the Nazis did. Nazi eugenics, like the whole of the eugenics movement, was a war on the weak, an attempt to target the genetically unfit rather than introduce genetic fitness into a population. Let's look at reproductive autonomy now. Reproductive autonomy is your right to determine how, when, and whether to have children. It is now a commonly accepted principle in medical ethics. If eugenics is a failed attempt at social control of reproduction, reproductive autonomy is the now accepted idea that individuals do have the right to control their reproduction. The history of this idea parallels the history of eugenics and interacts with it in strange ways. Sometimes the two are opposed, and sometimes the same individuals are promoting both. Now, efforts by individuals to control their reproduction by abortion and birth control go as far back as recorded history. Elixirs made from the pennyroyal plant were widely believed to induce abortion in medieval Europe, although such concoctions can actually kill you, and I don't recommend using them. If you look back to the time the United States was founded, however, you don't see much effort to control reproduction. Like most agrarian societies, American families typically wanted as many children as possible. Abortion only came up in the case of out-of-wedlock births, and, this is important, abortion was legal up to the time of quickening, the time that the mother can feel the baby move. Starting in the 19th century, however, there was a movement by women to start taking more control over their fertility. Childbirth was, after all, a very dangerous activity. At first, this was promoted by the simple assertion of a woman's right to turn down her husband's request for sex. It quickly evolved, however, in the 1830s into growing access to methods of reproduction and birth control. Advertisements like this one offered drugs to restore blocked menstruation, which was a widely known euphemism for abortion. Most of the anti-abortion laws that were on the books until Roe v. Wade were actually enacted in response to this rise in access to abortion and birth control. Despite this backlash, efforts to promote the right of women to control when and whether to have children continued. Margaret Sanger founded the first birth control clinic in the U.S. in 1916 and would later found Planned Parenthood in 1942. The current idea of reproductive autonomy has its roots in this birth control movement pioneered by people like Sanger. Already, though, we can see some of the strange interaction between the ideal of reproductive autonomy and eugenics. Sanger viewed herself as an ally of eugenics. She felt that if women had control over how many children they had, poor women in particular would choose to have fewer. This is also the outcome the eugenicists wanted, because they viewed the poor as unfit to breed. So reproductive autonomy rose out of the 19th and 20th century birth control movements. The horror of the Nazi atrocities and the global U-turn on eugenics, however, did not hurt the idea of reproductive autonomy. It strengthened it. The reaction to Nazi medical experimentation was the beginning of the doctrine of informed consent. And when people began to see eugenics as a mistake, they felt that the worst crime inflicted in the United States was forced sterilization, a violation of reproductive autonomy. By the time you get to the 1970s, patient autonomy is an established idea, and with that comes reproductive autonomy. The rise of reprogenetics, again, changes the relationship between reproductive autonomy and eugenics. Many view reprogenetic technology as the return of eugenics. Defenders of reprogenetic technology often appeal to reproductive autonomy. They say, no one is advocating coercive measures here. We just think that parents have the right to use these methods if they want to. We will return to this idea at the end of the presentation. IVF, the enabling technology. In vitro fertilization is the enabling technology behind everything we're doing here. This should be familiar to a lot of people by now. The first IVF baby, Louise Joy Brown, was born 33 years ago. Since then, 4 million infants worldwide have been born by IVF. 1% of all births in the United States are by IVF. The process is easy enough to describe. You mix sperm and eggs in a test tube and then allow the embryo to grow to the blastocyst stage, and then you implant it in someone's uterus. Although this sounds simple, the difficulties should not be underestimated. To harvest the eggs to begin with, a woman has to take a great deal of fertility medications, so she hyperovulates. She releases a whole bunch of eggs all at once. It used to be that the eggs had to be removed surgically, But these days they can do it using a hollow needle inserted through the wall of the vagina and guided by ultrasound. Once the eggs are harvested, there are difficult decisions to make. Not all of the attempts to fertilize an egg are going to be successful. If you don't fertilize enough, you might wind up without enough embryos. If you try to fertilize too many, you might wind up with leftover embryos. The excess embryos have been a source of controversy, as we shall see, because they are either thrown out, used as a source of stem cells for medical experimentation, or just sit in limbo indefinitely. The same issue comes up again when you have to implant the embryos. Not all of them will succeed in implanting in the uterine wall. If you don't try to implant enough, you might wind up without any babies. If you try to implant too many, you will wind up with a multiple birth, like 8, which we shall see is again a source of an ethical controversy. Safety issues. In 2010, the CDC estimated a success rate of 30.5% for IVF. If you go online, though, it isn't hard to find IVF clinics advertising success rates of 50 to 75%. Some studies indicate higher rates for certain birth defects. This might be caused by the large number of multiple births and the associated low birth weight. The baseline rate for having any birth defect is about 4%, but we don't have a comparable number for IVF babies. The number of embryos implanted per cycle by American clinics has been falling. In 2008, 87% of U.S. fertility treatments implanted 3 or fewer embryos per cycle. In 1999, that number was only 63%. In the United States, IVF is almost entirely unregulated. In particular, there are no laws regarding the preservation or destruction of embryos, how many embryos should be created as a part of a fertility treatment, or how many embryos should be implanted. The CDC monitors U.S. fertility clinics and issues an annual report making recommendations for best practices, but there is no requirement that these be followed. There is also an industry-run group called the Society for Assisted Reproductive Technologies, SART, which issues guidelines in an attempt to show the public that the industry can self-regulate. Most fertility clinics do follow SART guidelines, but if you want to find someone who will go against them, you can. SART limits the number of embryos you can transfer based on the age of the mother. For a woman under 35, they say only 1 embryo should be transferred. This did not stop Michael Kamrava from transferring 8 into the uterus of Nadia Suleiman, resulting in the first birth of octuplets, where all 8 lived. The case of Nadia Suleiman is discussed on pages 331 to 334 of your text, where Munson actually gets the name of the doctor wrong. He says the doctor was T.N.C. Chu. There was a Chu who was interviewed by the New York Times about the Suleiman case, and things might have gotten confused in editing. Munson also leaves out a couple important details in the case. First, it came out that Kamrava actually implanted 12 embryos. Second, he lost his license because of this and the publicity surrounding it. Some might argue the fact that Kamrava lost his license shows that the system of regulation works, the current system of regulation. On the other hand, one case where an egregious, high-profile violation of the guidelines gets punished hardly counts as a rigorous enforcement system. There is, however, a nice contrast in the regulation in Germany. Because of the legacy of the Nazi medical experiments, Germans are very sensitive to medical ethical issues, so they created in 1990 something called the Embryo Protection Act. This is all one long word in German, which I'm not going to try to read. The Embryo Protection Act allows for much closer regulation of in vitro fertilization. It is illegal to create more than 3 embryos at a time. This means that German clinics are not producing the large numbers of extra embryos that the American clinics produce. These extra embryos have become famous in the stem cell debates. It is illegal to alter embryos for reasons other than their own benefit. This rules out a lot of the fancy genetic experiments that go on in the U.S. It is illegal to create embryos unless you're going to implant. You can't create embryos just to experiment on it. And it is illegal to create an embryo that is a genetic copy of a living person. Thus, it is illegal to clone. It is worth emphasizing how much of a game-changer IVF technology is. Consider the difference between old-fashioned reproduction and what we might call newfangled reproduction. In old-fashioned reproduction, 2 people fall in love, or, well, maybe not, and decide to have a baby. Okay, well, half the time it's unplanned. So they have sex, and 9 months later, baby. This system basically gives you 2 chances to intervene in the process. You have contraception and abortion. Notice also that in each case, all you can do is stop the process. You can't change how it goes along. Now consider reproduction post-IVF. By my count, this process offers 9 chances to intervene, and the intervention isn't just a matter of stopping the process. You can alter how the process goes forward. In particular, you can make genetic alterations. If you have sperm and egg in a laboratory, you can select the exact sperm and egg you want. For instance, you can only select sperm carrying an X chromosome, guaranteeing that the baby will be a girl. You can also genetically alter the sperm or egg, which might be tempting if you assign moral status to the embryo and believe it shouldn't be tampered with. Once you mix sperm and egg, you can decide which embryos to culture, to allow to grow. And when they get to the 8 cell stage, there is another good opportunity to alter them genetically. Then you can decide which embryos to implant. And you get to decide whom to implant them in. As the healthy embryos grow, you can practice selective abortion. And finally, this all leads to the birth of a healthy stick figure baby. What this means is that a process which had previously been mostly random and unplanned is now under thorough human control, if we want to exercise it. This could mean a big change in human society. To see why, we need to look a bit more at the forces driving this technology. The change in human reproduction is not driven by some oddball desire to take the sex out of reproduction. All of these technologies developed to treat and prevent disease. The 2 most important categories of disease that have driven us to acquire the knowledge we now have are genetic defects and congenital defects. Genetic defects are problems arising from mutations or an error in copying genes. Once present, these defects can be inherited. Congenital defects are problems present at birth stemming from mistakes in fetal development. These are not inherited unless they have a genetic basis. So here are some examples of genetic and congenital defects which often come up in the context of medical ethics. First of all, we have single gene diseases. These are diseases that are caused by a change in a single gene and are inherited in a Mendelian fashion. This includes Tay-Sachs disease, Huntington's, cystic fibrosis, and sickle cell disease. There are also diseases that have a genetic component. For instance, the hereditary variety of breast cancer has 2 genes associated with it that partially determine whether or not you will get breast cancer. In fact, all diseases seem to have some level of genetic input. In addition, we have congenital defects. These include neural tube defects like anencephaly, microcephaly, and hydrocephaly. We've seen all of these previously. This also includes Down syndrome. So, in addition to research driven by desire to cure genetic and congenital defects, there is a lot of money simply in treating infertility. Between 12 and 15% of women ages 15 to 44 are seeking fertility treatment. That statistic is from Munson. Hopefully there aren't many 15-year-olds in that group. A round of IVF costs between $12,000 and $15,000, and you can spend up to $200,000 trying to get pregnant. This is a big deal. A final major benefit that has come from our drive to understand human reproduction is stem cell therapy. An important part of understanding how human beings develop has been understanding how cells differentiate, and this has led to an important spin-off technology of reprogenetics, the emerging use of relatively undifferentiated cells to heal damaged tissue and cure disease. This is called stem cell therapy, and we will look at it shortly. IVF takes reproduction out of the bedroom and into the lab. Well, what can we do once we are making people in a lab? One thing we can do is to sort and screen people by their genes. This is actually something we didn't need IVF to do. In fact, we've been doing this since long before IVF was possible. But the possibilities of this become much greater with IVF in place. So let's take a look at genetic screening and genetic testing. Genetic testing is the testing of an individual for signs of a genetic disease or trait. Genetic screening is a testing of a large population for signs of a genetic disease or trait. Both of these practices have been in place for a long time. The first major program of genetic screening in the United States was for the disease phenylketonuria, PKU. PKU occurs when a gene that produces an enzyme needed for metabolic processing is missing. Without this gene, a chemical called phenylalanine builds up in the blood, causing mental retardation. However, the disease can be basically controlled by putting the child on a special diet up to age 6. In 1962, a cheap blood test for PKU became available, and it became possible to screen all newborn babies for the disease and put infants with the disease on a special diet right away. This case of genetic screening seems uncontroversial. With this information, we can avoid a major hardship for children and parents. As a result, PKU screening is widely accepted. When my son was born, the nurses kept popping in to take blood and run tests. At one point I asked, "So what's this blood test for?" "PKU," the nurse replied. "But I've already done that," I said. "But you've already done that," I said. At that point, the nurse confessed that whenever they take blood from a baby, they tell the patients it is for a PKU test because parents automatically accept it. See the section in Munson on truth-telling in medicine for a discussion of practices like this. As we shall see, not all genetic screening programs have this kind of acceptance. Giving a PKU test to all newborns as genetic screening Giving a PKU test to all newborns counts as genetic screening because all babies are subject to it. This should be contrasted with testing that can be done to the individual at their discretion. A good example of this is the test for Huntington's disease. Huntington's is caused by a single dominant gene, so you only need one copy of the gene to get the disease. And if your parents have it, you have a 1/2 chance of inheriting the disease. The disease starts to take hold when you are in your 40s or 50s. It begins with little shakes and tremors, but then progresses to a complete loss of motor control and mental disorientation. It is always fatal, and there is no treatment. The gene for this was discovered in 1993, and a test quickly developed. So here's the question. If you thought you might have this disease, would you want to be tested? If you were planning on having children, would you be obligated to be tested? Another big complication with genetic testing and screening comes from genetic discrimination. If your genetic profile is public knowledge, people will deny you opportunities based on this information. The two biggest groups of people who engage in genetic discrimination are insurance companies and employers. Insurance companies have denied coverage to people because of their genetic profile. They have even announced that people with a genetic predisposition to a disease have a pre-existing condition and thus were not entitled to coverage for that disease. This has all led to widespread cases of people avoiding needed medical tests to keep insurance companies from finding out the results. Employers have also engaged in genetic discrimination. They have denied jobs to people who are very likely to get sick. In the case of sickle cell disease, people were denied jobs just for being carriers of the gene. A lot of these problems will hopefully be fixed by the Genetic Information Non-Discrimination Act, or GINA. GINA prohibits employers from requiring or requesting a genetic test, purchasing genetic information, or disclosing genetic information. GINA prohibits insurers from requiring or requesting genetic information, using genetic information to determine eligibility or premium, and from disclosing genetic information. I think this makes it clear that genetic testing and genetic screening are fraught with ethical issues. There's more detail in your text, both about the kinds of disease that are tested and screened for and the ethical issues raised by all of them. Here I will just hit a few highlights. For genetic screening, how do we deal with the potential for genetic discrimination from insurers and employers or the general public? How can we best keep genetic information private? Can we, when we engage in genetic screening, target certain populations? Can you make genetic screening mandatory? Ethical issues in genetic testing. Is there a duty to know your genetic profile to help your family members, especially your children? Is it reasonable not to want to know your genetic profile? Is there a duty not to have biological children if your genetic profile indicates severe problems? Preimplantation genetic diagnosis. If we combine IVF with genetic screening, we get a technique called preimplantation genetic diagnosis. In IVF, embryos are already selected for their ability to implant in the uterus. While you are at it, you can also select them on the basis of their genetic characteristics. PGD can be done with infertile couples as a part of ongoing IVF fertility treatment, or it can be done with a fertile couple that decides to use IVF instead of the regular method, because that will allow them to pick the embryo that they want. Some of you may have seen the movie Gattaca with Ethan Hawke and Uma Thurman. The movie was about a future in which everyone is genetically engineered. In that movie, the genetic engineering techniques they used to create everyone, to create Uma Thurman, is PGD. Currently, PGD is used to prevent genetic disease, including Tay-Sachs, Huntington's, and cystic fibrosis. For instance, if you and your spouse are both Tay-Sachs carriers, 1/4 of your embryos are doomed to die painfully before age 5. So why not just do IVF and not implant those? PGD has also been used to create siblings that will be a good tissue match for an existing sibling. These are so-called survivor siblings. We will look at this more at the end of the presentation. Genetic therapy and genetic engineering. PGD allows us to sort through existing embryos by their genotype. Why not take things a step further and actually alter their genes? This is something that we can do either as adults or to embryos in the lab. And it is something we can do to either treat disease or actually make healthier people even better. It used to be that all this stuff was referred to as genetic engineering, and in fact people still use that term when discussing the genetic alteration of plants, animals, and microbes. However, when we alter human genes to treat disease, people now refer to it as gene therapy, which sounds much less sinister and Frankenstein-y. People also tend to refer to all forms of genetic alteration in humans, whether to treat disease or to enhance, as genetic intervention. It will help to get clear then on the kinds of genetic engineering and genetic alteration which are conceivable. The first distinction we need to make is between genetic therapy and genetic enhancement. Gene therapy is the alteration of DNA or patterns of DNA activation to cure disease. We have already seen that there are a wide variety of diseases that are caused by a single gene. And if we can change that gene, we can treat the disease. This is called gene therapy. It essentially takes someone who is below normal functioning for some trait and returns them to normal. We could apply these same techniques to the genes of a healthy person, though, to make them even better. For instance, we could genetically alter the hemoglobin of an athlete so it is better able to transport oxygen, just thus giving the athlete more strength and endurance. This would be called genetic enhancement. It takes a trait that is already within the normal range for the species and makes it even better. So we will use the terms genetic intervention or genetic engineering to refer both to gene therapy and genetic enhancement. Another important distinction we need to make is between germline and somatic cell intervention. Germ cells are sperm or eggs or any tissue that produce sperm or egg and thus contribute to their DNA. These cells are important because their genes are the ones that will be passed on to the next generation and the generation after that and so on. These cells are often referred to as the germ line because they form a continuous line of mother and daughter cells that runs on through the families of people. A somatic cell is any non-germ cell. These are the cells you can change without affecting future generations. Somatic cells make up most of who you are, but from a genetic perspective, they are just temporary outgrowths designed to protect the germ line as it passes through generations. Germline intervention is then an intervention that alters germ cells. These cells will be passed on to every future generation. This can happen if you alter an embryo at the 8-cell or blastomere stage. At this point, there is no differentiation between the somatic cells and the germ cells. So, if you alter DNA here, it won't just be affecting the embryo. You are affecting that embryo's future children, grandchildren, and great-grandchildren. Somatic cell engineering, on the other hand, only affects the somatic cells and thus can't be passed on to future generations. Suppose you have an adult, for instance, with cystic fibrosis. This means that they've got a genetic malfunction in all of their cells that causes the cells to ooze a lot of mucus. When your lung cells ooze this mucus, it fills up your lungs and makes it very difficult to breathe. Well, if you try to genetically alter the tissue in your lungs so they aren't producing as much mucus, you are doing somatic cell engineering. You are not doing anything that will affect your children. This is a hypothetical procedure, but if you did it, it would only affect you. You put these distinctions together, and you get 4 kinds of genetic intervention. And these 4 kinds of genetic intervention will have different kinds of ethical importance, all different levels of ethical controversy. The least controversial form of a genetic intervention is a somatic cell treatment. This would be genetic engineering to cure a disease, to restore functioning to species normal level, and do that in a way that only affects one person. Genetic treatment for cystic fibrosis or diabetes would fall into this category. You can also imagine somatic cell enhancement. This would be engineering someone to improve their abilities beyond what is normal for the species. If you had a regime where you took an athlete's bone marrow and genetically altered it to produce superior blood, this would be somatic cell enhancement. This is going to be more ethically problematic than somatic cell treatment in many people's eyes. In sports, it is called gene doping. It is banned by the World Anti-Doping Agency, but there is no way to test for most forms of it. It also seems likely to me that at least some of the world's militaries are experimenting with somatic cell enhancement. There are dozens of nations, including our own, with both the ability and the motive to push the boundaries of what their soldiers are capable of. Another more controversial kind of genetic engineering is germline treatment. Germline treatment would occur when you alter the genes in a way that can be passed on to all of your descendants, but you are doing it to prevent disease. The gene for Tay-Sachs has been a part of people's germline for generations. So why not just take that out of the germline? If you are fertilizing an embryo in vitro and remove the Tay-Sachs genes and implanted it, and grew it into an adult, you would be sparing all the descendants the horror of Tay-Sachs. The most ethically controversial form of genetic engineering would be germline enhancement. This would be enhancing someone during the embryo stage so that it affects their germ cells, and doing this not to cure disease, but to improve the individual. So, if you did what you did to the athlete's blood cells, except you did it to an embryo, so that that embryo and all its future offspring would be more athletic, that would be germline enhancement. There are actually a whole bunch of ways to alter genes, but the most prominent way to do it in humans involves a virus. It is easy enough to directly insert the DNA you want into a virus, and after that, the virus will do what it is evolved to do, infect your cells and insert copies of the DNA, now including the DNA that you want to insert. You can also change the cellular environment to shut off or turn on specific genes. This may not seem like it would count as genetic engineering, but it turns out that this kind of alteration can be heritable. Although most people regard germline enhancement as unthinkable in humans, it is practiced routinely for almost every other species on the planet. You have probably eaten something today that is the product of germline enhancement. Most of the commodity crops grown in the United States, soy, corn, wheat, and rice, are all enhanced in their germline to do things like resist pesticides or produce their own pesticides. Fish are going to be the next big food you see commercially genetically engineered. Genetic engineering is also used in the production of many medicines and other useful chemicals. There are details about this on page 276 of your text. In the laboratory, basically everything has been subject to germline enhancement except human beings. Over to the right of your screen is Andy, our closest GM cousin. He is a transgenic rhesus monkey. He has had a jellyfish gene for fluorescence inserted. This is a common thing that scientists use to test genetic engineering techniques. There are jellyfish that glow in the dark. You can put that gene into various animals, and then it's very easy to see if you have successfully genetically engineered the animal. Typically, the whole animal won't glow in the dark, but if you take a tissue sample, that will glow in the dark. So you notice that Andy here has kind of a purplish hue to him. That's his jellyfish heritage. As far as human beings go, the only thing that has been experimented with publicly is somatic cell treatment. There may have been, as I mentioned, covert efforts at somatic cell enhancement in sports or within the military, but that's something we can only speculate about. Experiments with somatic cell treatment in humans have had mixed results. A detailed list of examples is on pages 240 to 244 of your text. These include success stories of treatments with various genetic malfunctions of the immune system, as well as Parkinson's disease, inherited retinal degeneration, and AIDS. On the other hand, there have been a number of high-profile deaths in these experiments. Teenager Jesse Gelsinger was suffering from a mild version of a metabolic disease that is usually fatal. In Gelsinger's case, the disease could be managed with diet and medication, but he enrolled to help those who have the more common fatal version of the disease. He died suddenly, apparently from a reaction to the virus used to deliver the genetic alteration. The story is given starting on page 123 of your text. There was also an experiment in France with children who had X-linked severe combined immunodeficiency syndrome, XSCIDS. This is more famously known as bubble boy disease because the babies are born without a functioning immune system, and so they have to live in a bubble to keep them free from disease. Gene therapy was showing progress here, but a couple of babies developed leukemia and died. Stem cell therapy. Stem cells have been a huge public issue for over a decade. Before 9/11, people were saying that the stem cell debate would be the defining issue of the Bush presidency. The debate on stem cells went nowhere, as people lined up on the same sides as they did in the abortion debate, and any political difference between the stem cell debate and the abortion debate was ignored. I think we are reaching the point, though, where advances in technology will make a lot of this debate moot. We are definitely heading towards a time somewhere in the future when the use of embryonic stem cells will no longer be necessary, because everything we need to do can be done with adult stem cells. Let's go back and review the basics of stem cells to see how this happened. The human body contains 120 different kinds of cells, more or less. But each cell has the exact same DNA. There aren't 120 different kinds of DNA in your body. There's only one genetic code for you. Your DNA is like a cookbook with 120 different recipes in it. Now, at some point in the development of any cell, one of these recipes gets marked. The corner of the page gets folded down so the cell thinks, read this recipe only. That's why that cell becomes a heart cell or a liver cell or a brain cell. Stem cells are cells that do not have one of their recipes marked yet. So they are flexible. They can become any kind of cell, and this gives them remarkable healing power. Stem cells are divided conventionally into 3 levels of power. A totipotent stem cell is a cell that can become any kind of cell. The only totipotent cells are in the original embryo, when there are just a handful of cells that are completely undifferentiated. A pluripotent cell is one that can become most kinds of cells. The one kind of cell it can no longer become is the placenta. At first, it was believed that the only places to find pluripotent stem cells were in the early development of the embryo, when it has about 100 to 300 cells, and also in the germ cells of a later developed fetus. Now it looks as though we are capable of taking any kind of cell and restoring it to its pluripotent state, unmarking the recipes in the cookbook, as it were. The only recipe that we can't access again is the placenta recipe. Multipotent stem cells are cells that can become only a few kinds of cells. These are the stem cells that are known to be at work in your body right now. So in your bone marrow, you have what are called hematopoietic stem cells. These can become any kind of blood cell, and the most well understood kind of multipotent stem cell. I said that stem cells had tremendous healing potential. The actual technology that people are seeking to develop is called stem cell therapy. Stem cell therapy is the process of implanting stem cells into ailing tissues in order to heal that tissue. So if you have brain tissue that is damaged from Alzheimer's or Parkinson's disease, you introduce the stem cell tissue, and it's like neural silly putty. It will latch on and replace the damaged tissues. And you can do this for weakened heart muscles, or for the islet cells in the pancreas which produce insulin. Another interesting application that is not as much talked about is the potential to create an unlimited supply of disease-free blood. If we really understood how hematopoietic stem cells, the stem cells that create blood cells, worked, we would create our own little bone marrow factories that would produce blood products cheap. That would be very useful. Right now, though, most of these technologies are speculative, and the only thing that is really being done with stem cells is just research. And the research that is being done is basic research. The reason scientists are fascinated by stem cells is that they want to find out how the whole process works. It's a big, exciting mystery. How is it that a cookbook of 100 different recipes gets marked for one recipe? This relates to some fundamental issues in developmental technology. And that's what's exciting for scientists. So, although the medical potential is what is most advertised, most scientists are motivated by plain and simple curiosity and the prestige that comes with solving a fundamental mystery. The problem up until now. Up until now, researching all these potential technologies has been controversial because of where the stem cells come from. I mentioned previously that pluripotent cells were thought to only come from embryos and fetuses. I want to go into this in a bit more detail here, because there are a wide variety of sources of stem cells. But up to now, the rule has always been, the better the cells, the more controversial the source. The most famous source for stem cells is the human embryo. However, there are a variety of ways to get embryos, each with its own ethical problems. The original focus of the stem cell debate was on embryos left over from IVF fertility treatment. As we mentioned previously, people generally create more embryos than they implant. These leftover embryos generally wind up in a sort of limbo. They are sometimes destroyed, but often couples are reluctant to do that. They also typically don't want more children, so the embryos just sit in freezers, tens of thousands of them, and I don't think anyone has a good sense of how long they will keep. In any case, it is often thought that these embryos are never going to be implanted. So, why not use them for research? There are other ways to get embryos, though. Some people thought that creating embryos specifically to be implanted would be less objectionable than using IVF embryos, but that idea didn't really fly. People also sometimes talk about using cloned embryos. If the goal is ultimately to transplant stem cells to a patient, you are going to want the stem cells to be a genetic match for the patient. And the best way to do this would be to use an embryo cloned from the patient. We will talk about cloning more in the next section, and we will also talk later about parthenogenesis and human-animal mosaics. All of these have been ethically controversial. A final source that is worth knowing about for germ tissue is aborted fetuses. Although for the most part cells lose their pluripotency as the fetus develops, the germ cells, the cells that will later create sperm and eggs, are still pluripotent. One of the most famous stem cell researchers, John Gearhart, does all his research on tissues taken from fetuses that had to be aborted to save the life of the mother. So that's where we thought we could get pluripotent stem cells. There are also less controversial sources of stem cells, but these were originally only thought to be multipotent. These include cells found in adult bone marrow, intestine, brain, and maybe also. It also includes umbilical cord and placental blood. In addition to these being only multipotent, these were not as good because they tend to carry mutations. As we walk through this vale of tears, our cells mutate. The body tries to protect the germ line from mutation, but the older you get, the more your somatic cells mutate, which is ultimately what can give you cancer. It is worth reminding ourselves about the nature of the embryos we are dealing with here. I discussed this in the Human Development section of Presentation 10 on abortion, but I want to go over it again. We are looking at embryos between the 3rd and 14th day after fertilization. These embryos have 1 to 300 cells formed into a hollow ball called a blastocyst. Inside the ball is a mass of about 20 cells called the inner cell mass. These are the pluripotent stem cells, which will later form the fetus. The outside of the ball will form the placenta. To get pluripotent stem cells, it was thought you would have to crack open the shell, open it, and take out the inner cell mass, destroying the embryo. But maybe that isn't true. We can now add a new kind of stem cell to our vocabulary: induced pluripotent stem cells. These are adult cells that have been reset to their pluripotent state. It has long been believed that these were possible, At the very least, the science has always been heading here. The goal of stem cell research, after all, has been to understand the process of differentiation. And in science, the ultimate sign of understanding something is being able to control it. Knowledge, as Francis Bacon taught us, is power. In the kind of science we are doing, you can tell you have knowledge and power if you have identified genes. And for induced pluripotent stem cells, genes have been identified. Shinya Yamanaka and Kazutoshi Takahashi at Kyoto University have identified 4 genes that, when altered, restore the cell to its pluripotent state. Their experiment has been replicated by 2 other groups, which is another crucial sign of knowledge and power. James A. Thompson, a famous stem cell researcher at the University of Wisconsin, has discovered a separate recipe for restoring adult cells to their pluripotent state. This one involves a different set of 4 genes that overlaps with Yamanaka and Takahashi's set. There have also been a lot of experiments which show that cells can be induced to switch type. Bone marrow cells have been used to produce heart cells. Non-insulin producing pancreas cells have been converted to insulin producing ones. In the long run, the ability to turn adult stem cells into pluripotent cells will make the use of stem cells from controversial sources unnecessary. This is also where everyone wants to head, regardless of how they feel about the use of embryonic stem cells, because induced pluripotent cells will have so many advantages over embryonic cells. They will be cheaper and easier to acquire, and since you take them from the patient who needs the therapy, you can easily get cells that are a genetic match without having to go through the complicated process of cloning. There is a long road from here to the bright induced pluripotent future, however. The same genes that induce pluripotency can also create cancer cells. In general, the boundary between cancer cells and stem cells is pretty porous. They are both kinds of cells that proliferate indefinitely. The process of inducing pluripotency needs to be mastered, and until it is, many researchers feel they will need to work with stem cells from morally controversial sources, like embryos and aborted fetuses. In fact, there may be no way to get to the point where human embryonic stem cells are unnecessary without first fully understanding human embryonic cells, and that means experimenting with them. And even if we master stem cell therapy with induced pluripotent cells, there will be more and stranger moral problems on the horizon. If we can induce pluripotency, we might be able to induce totipotency. A totipotent cell, remember, is like a zygote, the original fertilized egg. It is all you need to make a human being. We saw in the abortion section that many people attribute moral status to the zygote because of its potentiality. It has, as Marquise said, a future like ours. Well, if you can induce totipotency, then any cell in your body now has the potential to be a new human being. The cell would need to be reprogrammed and implanted in someone's uterus. But there are a lot of things that need to happen before a zygote can become a fetus too, and the odds were never as good as they are after implantation. Now, it really seems like we're in the realm of science fiction. There are actually a lot of very exotic technologies on the outer edge of reprogenetics. Let's look at another popular one. Cloning. Cloning, more properly known as somatic cell nuclear transfer, is the creation of an embryo that is a genetic copy for an existing organism. IVF has given us the ability to grow embryos in vitro. Once we can do that, we can start creating embryos by other means, culturing them in vitro, implanting them in a uterus, and allowing them to grow. And that's what we do with cloning. Let's start by remembering the important distinction from the section on genetic engineering between germ cells and somatic cells. Germ cells are sperm, eggs, or any of the cells that create sperm or eggs. These germ cells form an ongoing line from parent and child cells that ultimately links all life on Earth. Somatic cells are any kind of non-germ cell. These are the body cells produced seemingly as side effects of the germ line. Your body grows up and around the germ line, protects the germ line, and passes the germ line along, but it doesn't change the germ line. Somatic cell nuclear transfer is the creation of a viable embryo that is a genetic match for an existing organism by replacing the nucleus of an egg with the nucleus of a somatic cell. Here are some facts to remember. I think these days most people are more aware of these facts than when cloning first started appearing in the news, but they still bear repeating. A clone is not a Xerox copy of someone. Clones do not appear fully grown like they do in Star Wars. Cloning is not some kind of human photocopier. Cloning is a way of making a baby. Clones are about as identical as twins. They share the same DNA, but were not raised in the same environment. Another way of thinking about a clone is that a clone would be your delayed twin. You've always wanted a twin sister, now you can have one. She will just be a couple of dozen years younger than you and perhaps born of your womb. Okay, so here's cloning in 3 absurdly easy steps. You get an egg, and you get a somatic cell, like a skin cell. The nucleus of the egg is haploid. It has half the usual number of chromosomes. The nucleus of the somatic cell is diploid. It has the full set of chromosomes. Next, you remove the nucleus of an egg. This is a picture of a real live nucleus being removed from a real live egg. Next up, you replace the nucleus of the egg with the nucleus of the somatic cell, and suddenly you've got an egg with a full set of chromosomes. Now, if you bathe the egg in some chemicals and give it a little zap of electricity, you can convince it that it has been fertilized. And then it starts dividing, and the result is a blastocyst. There are many things you can do with cloning, and it's important to distinguish them. Reproductive cloning is cloning for the sake of producing a living organism. So at the end of that last process, you get a blastocyst. If you put that blastocyst in someone's uterus and grow it into a baby, you are engaged in reproductive cloning. Therapeutic cloning is cloning for the sake of harvesting the stem cells. Here, you do the previous process. You get your blastocyst, but then you break it open and you harvest the stem cells, either for research or perhaps someday for the sake of stem cell therapy. All right. There's another technique that gets less attention that's very similar to cloning. It's called parthenogenesis. In parthenogenesis, you catch an egg that's still at the stage when it has all 46 chromosomes inside it. This is because it has something called a polar body inside it still. You can actually trick this egg into thinking it has been fertilized. This is a quick way of getting yourself a zygote and then an embryo on down the line. There are some animals out there, some lizards and some fish, that actually reproduce parthenogenically on their own. It is conceivable that we could get a parthenogenic embryo to grow at least as far as the blastocyst stage. Let's turn now to the stuff that can really make people think scientists are creating monsters. Mosaics, chimeras, and other hybrids. Right now in the laboratory, it is fairly common to mix human and animal tissue. Such mixed cultures get called mosaics or chimeras or hybrids. The easiest way to do this is to simply fuse the embryos of different organisms prior to individuation. One of the things I emphasized when discussing human development is that in the abortion presentation is that prior to implantation, you do not have a fixed number of individuals. If you cut an 8-cell embryo in two, you will get 2 embryos, and this is one way to get twins. More interestingly, if you moosh 2 8-cell embryos together, you will get 1 embryo. Sometimes twins will form and then merge back together. You can even moosh together 2 embryos created by a different sperm and egg. So sometimes you get fraternal twins, twins made from different sperm and eggs, mushed together in one baby. The resulting person will have different DNA in different cells. There was an episode of CSI where they were unable to nab the perpetrator at first because he was a chimera and his DNA came out differently on different tests. Although CSI is not a very realistic show, such cases do exist in humans and are even more common in animals. There are even cases where male and female embryos are mooshed together at the 8-cell stage, and the resulting person has a Y chromosome in half their cells. How they develop sexually depends on which set of cells winds up dominating their reproductive systems. Intersex human chimeras can even be fertile. Well, nature can work wonders, and in the lab we are just beginning to catch up. We can moosh together embryos of different species, and in some cases grow that into an adult. The animal pictured here is a geep. In 1984, people made a geep by mixing the embryos of a goat and a sheep. This is called a mosaic, because the cells that move through the developing fetus are a mix. These days it is quite common for human-mouse mosaics to be made for the purpose of tracking the development of cells in the embryo. Which cells wind up where, for instance. I read an article a while back about some research into autism, which involved growing human neural cells in frog embryos. The idea was that the human tissue would respond differently to electricity than the frog tissue. They then compared the neural tissue from normal people and autistic people to see if they would function differently in frog embryos. No embryo like this is ever grown to adulthood, nor could it be. They are just not viable. However, they are very useful in laboratory experiments. Needless to say, this touches directly on ethical issues of naturalness because we are crossing species boundaries. We are creating human-animal hybrids. This is very island of Dr. Moreau, except that it happens at the microscopic level. Another possibility, and this might even be creepier for some, is that we could actually take a nucleus of a human cell and put it in an animal egg. I think the only reason to do this is just that animal eggs are easier to get than human eggs. But this would also create a human-animal hybrid of a sort. Again, none of these embryos last. You cannot create a person or any other kind of creature out of them. Anything that will grow or develop. But you will have human-animal hybrid tissues cultured in a lab, and this can be done for the research purposes or for the creation of stem cells. Surrogacy. So let's review the process of artificial reproduction. We start with sperm and egg, and we can screen and sort these genetically. We can also create zygotes by somatic cell nuclear transfer or parthenogenesis. These can be grown in vitro to the 8-cell stage, where we can create chimeras, or we can engage in germline treatment or enhancement. We can continue to grow the embryo to the blastocyst stage and harvest the stem cells. We can sort the blastocysts for pre-implantation genetic diagnosis. Finally, we can choose whose uterus we decide to put the blastocyst in, including someone who is not genetically related to anyone involved in the process up to this point. This is called gestational surrogacy, and it raises its own set of ethical issues. Gestational surrogacy occurs when one woman agrees to carry a baby for another. The surrogate may or may not also provide the egg. This is not cutting-edge technology anymore. It happens all the time, but it raises some ethical issues. First, what if the surrogate decides she wants to keep the baby? This is discussed in your textbook in the Calvert and Baby M cases. In the case of Baby M, the surrogate was also the biological mother. This wasn't even a high-tech procedure. The surrogate, Mary Beth Whitehead, simply agreed to be artificially inseminated by the husband of the infertile couple, whose name was Stern. Honestly, it isn't that different from what happened in monarchies of days gone by, when the king impregnates a concubine and declares the child heir to the throne because the queen is infertile. In any case, Whitehead decided she wanted to keep the child. The case made it up to the New Jersey Supreme Court, which ruled that the surrogacy contract was invalid and that Whitehead was the legal mother of the child. Nevertheless, primary custody was awarded to the Sterns because the judge felt that they would provide a better home environment. In the Calvert case, the surrogate was not the genetic mother of the child and so had less claim to the child, and in this case, the courts denied all her parental rights. Another issue that comes up with surrogacy is whether you can pay someone to be your gestational surrogate. Some people feel that this is a form of selling your body, like prostitution or organ sales. Other people feel that it is a form of baby selling or human trafficking. Some states have simply outlawed surrogacy. What's more common is for states to ban paying surrogates for anything besides their medical expenses. So let's look at ethical issues that span all of reprogenetics. Here again is the whole process of newfangled reproduction. We are able to intervene throughout this process. Most of these interventions will either take the form of selection or alteration. We pick the thing we want to contribute to the baby, or we alter it the way we like it. In either case, certain issues will come up repeatedly in the process. At any stage in the game, we can distinguish between germline and somatic cell interventions, for instance, and we can distinguish between treatment and enhancement interventions. I want to close by talking about a few other issues that come up at every stage in this process. I want to talk about sex selection, save your siblings, and consumer eugenics. One issue is sex selection. There are many opportunities to choose the sex of your baby. You can simply sort sperm, for instance. The ones that carry an X chromosome are much heavier than the ones that carry a Y. You can also select for sex during PGD. Many U.S. fertility clinics offer this service, as do some online hucksters. In India and China, it is common to simply use ultrasound to identify female fetuses and abort them, and this has led to severe gender imbalances. India and China are estimated to be short 160 million women and girls. This is equal to the entire female population of the United States. This is also happening in some of the former Soviet republics, which developed relaxed attitudes towards abortion during the communist years. This map of sex ratios in China is from the journal Nature. All the areas in white have a normal gender balance. You notice that this is mostly the sparsely populated western desert and occupied Tibet. The dark areas have as many as 3 males for every 2 females. This gender imbalance has led to increased human trafficking, mostly in the form of bride buying and forced marriage. It also leads to a simple increase in the crime rate. Young men are frustrated because they lack the social status which comes from being the head of a family, and they are simultaneously freed from the responsibilities of family, and this leads people pretty naturally into crime. One thing that you can do with this technology is ensure that your next child is a tissue match for an existing child who is in need of a bone marrow transplant. Sometimes when a child has a genetic blood disease or leukemia and needs a bone marrow transplant, sometimes when a child has a genetic blood disease or leukemia and needs a bone marrow transplant, they cannot find a donor who is a match, the parents decide to have another child or to have another child sooner than they originally planned. You can do this the old-fashioned way and simply roll the dice, but often, if you are dealing with genetic problems, the new child could also have the disease. So parents choose to use PGD to ensure that their next child will have the right genes to save their sick child. On page 348 of your text, Munson discusses a case like this, where the parents of Anissa Ayala decided to have another child to give Anissa a bone marrow transplant. This actually required the father to get his vasectomy reversed. The Ayala's case hit the media, and they were inundated with criticism. Many people seemed to think that the new child would be killed for her organs, or that she would somehow not be as loved as the Ayala's other children. More charitably, people thought that a decision like this would be a slippery slope to a situation where people are killed for their organs or created and not really loved. In any case, Anissa Ayala and her younger sister Marissa lived happily ever after, and Munson estimates that about 2,000 savior siblings have been born in the U.S. Okay. We began this presentation by talking about the eugenics movement, which culminated in the Nazi T4 euthanasia program, where 275 people with disabilities were murdered. Reprogenetic technologies now give us a level of control over the inborn faculties of man that the eugenics movement dreamed of. Disabled people are obviously concerned about how this technology will be used. The failure of the eugenics movement makes a large-scale government-led attempt to control the gene pool very unlikely. However, the success of the reproductive rights movement has enabled what seems to be another sort of eugenics, one driven basically by consumer choices. Reproductive autonomy is now an established principle of ethics. People in the industrialized Western world basically agree that you have the right to determine how, when, and whether to have children, although the exact extent of this right is debated. This has enabled individual couples to take advantage of reprogenetic technology to shape the inborn qualities of their children. In the 20th century, they sterilized people with disabilities. Today, couples at risk of having a child with disabilities can take steps to simply avoid having a child like that. But the situation now is very different than it was in the eugenics era. In addition to talking about the reproductive rights movement and the eugenics movement, we can talk about a third movement, the disability rights movement. For most of the 20th century, If a child was born with disabilities, the family would shut it up in an institution and never speak of it. These institutions produced a lot of horror stories about inhumane and cruel treatment. The story of Baby Owens, where the mother refers to her child with Down syndrome as mongoloid and refuses the child even basic medical treatment, is a remnant of this era. Starting in the 50s, though, people worked to reform the institutions for the disabled, and then later to reintegrate the disabled into society. And it turns out that many people with disabilities were capable of much more than people had thought. Down syndrome is a really amazing example of this shift. The word mongoloid now sounds antiquated, like Chinaman or Negress. And the hit show Glee has featured 2 characters with Down syndrome played by actors with Down syndrome. This change did not happen spontaneously. It was the product of organized activism by people with disabilities, their families, and their advocates. And it has resulted in important legislation, including the landmark Americans with Disabilities Act, which protects the disabled from discrimination and requires public institutions to provide the disabled with reasonable accommodation. This raises the important question: will consumer eugenics harm the disabled the way the old eugenics movement used to? On the surface, it seems like the answer is no. The crimes of the old eugenics movement were all based on coercion, forced sterilization, and ultimately rounding people up and murdering them. But consumer eugenics is founded on choice and the principle of reproductive autonomy. Many disability rights activists are still worried, however. When people take steps not to have a child with a certain disability, people who currently have this disability feel disvalued, even insulted. The message they hear is, "Your life is not worth living. We do not want to create another person like you." One of the basic messages of the disability rights movement has been that when people have a condition that prevents them from functioning in society, society must change to accommodate the individual, not the other way around. Will society continue to accommodate itself to the disabled when people are born? Will society continue to accommodate itself to the disabled when people can say when a disabled person was born, someone should have taken steps to keep this person from having ever been born? All of this raises a much deeper, more philosophical question, which I want to close with. What sorts of people should there be? We now have more control over what kind of people will fill up the world. It is less a product of chance or God, and more a product of design. So what do we do with this power? What sorts of people should there be is the title of a 1984 book by Jonathan Glover, which speculated on the impact of all sorts of wild technologies on what it means to be a person, from genetic engineering to direct mind-to-mind contact. It is also the title of a group blog that covers disability issues. The bloggers, as disability rights activists, seem to have an answer to their title question. They think that there should be all kinds of people. The advocates of consumer eugenics seem to think that there should be the best kinds of people. See the essay by Julian Savulescu in your text for an important example of this attitude. These answers are vague and simplified, and they certainly don't represent all the possible answers. So I just want to leave you with this as a final question to sleep on. What sorts of people should there be?